Today we're going to look at the process of replication when it comes to prokaryotes.
The first part of the replication process is called initiation. This is when the initiator protein binds to the origin of replication, causing a local unwinding and a short stretch of single-stranded DNA. After the replication process is initiated, then comes the unwinding phase. Helicase, as shown on the diagram, attaches at the replication fork and moves into the fork, breaking hydrogen bonds, creating single-stranded DNA. Single-stranded binding proteins coat single-stranded DNA to protect it and stabilize it and prevent it from being a double-stranded DNA. Then you would have DNA gyrase, which is a topsomer that relieves supercoiling ahead of the replication fork as it unwinds. It's shown in orange on the diagram. After the initiation and unwinding process, then comes the elongation process. Now, the elongation process happens on both sides of the fork, but for this example, we'll only look at the left fork, where our leading strand will go from a 5 to 3 direction towards the fork, and our lagging strand will go to a 5 to 3 direction away from the fork. First, an enzyme called primase synthesizes short RNA primers to allow DNA synthesis to begin. This can be seen in orange on the diagram. You can also see a free 3' OH created by the RNA primer. This is used for the DNA polymerase to start its DNA synthesis, since DNA polymerase cannot start from scratch. This is where DNA polymerase 3 comes in. DNA polymerase 3 synthesizes DNA and can be shown in yellow on the diagram. It starts off from the free 3' OH spot of the RNA primase, synthesizing DNA. It should be worth a note that replication occurs from the 5' to 3' direction on both strands. Because the two nucleotide strands of DNA are antiparallel, which means they're parallel but they move in opposite directions, one strand, the leading strand, has a continuous synthesis of DNA, while the lagging strand has fragmented synthesis of DNA. The short length of DNA created by the segmented synthesis of DNA by DNA polymerase 3 is called Okazaki fragments, which can be shown in yellow on the diagram. DNA polymerase 1, which can be shown in green on the diagram, cuts out RNA primer and uses 5' to 3' polymerase activity to replace RNA with DNA. DNA ligase fills in the nick between the DNA created by DNA polymerase 3 and DNA polymerase 1 without filling it with a nucleotide. DNA polymerase 1 is used to change RNA primers into DNA, and exonuclease activity is the proofreading of the DNA during replication. If DNA polymerase 1 is not functional at the time, then fragments are not going to be filled and proofreading will not occur, causing mutations in DNA, causing mutated proteins during translation. And there you have it, the entire DNA replication process.